I will spend the next 40 minutes. I hope I don't exceed the time, but I'll try to keep it in that time. Trying to tell you about some of the things we have done in our lab. Unfortunately, I may not talk about any biosensor work because I thought in the current scenario, it would be better to talk about drugs and drug resistance and our work on antibiotic resistance a bit, and how we as basic scientists search for problems. That's what I thought I would do. But then, as you can look at my title, it is got to do with more structure. What I want to impress here is that how structure is so important in our in trying to go forward in science. And I mean, this is a quote by James Watson, and it just tells you how important structure is. And without even saying, the structure of DNA, which you all loosely draw without even thinking, at one point, man did not know what it looked like. And just by being able to uncover the structure of DNA, for example, was the first stepping stone towards understanding biological structure. And then lot of new structures came over the years. Structure has paved the way for understanding both the processes of transcription and translation, and amassed many Nobel Prizes, because photography at the atomic level really makes you understand a lot of stuff. And here I am just, everybody is in the, engulfed by the coronavirus. And I'm just wanting to tell you that this is our coronavirus molecule here. And if you look, we are trying to search for antivirals and vaccines, and it has only 16 proteins. Okay, and one of the proteins, which is a protease, because it's formed as like one big polypeptide chain, and then it becomes 16 proteins via a proteolytic process, which is this protease. People are trying to find inhibitors, which are peptide-like analogs, to be able to use in the clinic. And already this has been taken up by Merck for clinical trials, because it's a need of the hour. So there are only 16 proteins, and yet it has bent the whole world. Now, I'm going to be concentrating mostly on bacterial resistance and bacterial drugs, and how do we go about searching for new drugs. And you can see there are a lot more enzymes, 5,000 or maybe 10,000 in humans. And yet, you know, we are still having so many diseases which we don't know. Now, look at a virus with 16 proteins. Look at us, or even bacteria, which is less complex, 5,000 or more proteins. Now this gives us more options. That is true. 5,000, you can kind of target many essential proteins in this. So, but what happens as a result of options? You can have side effects. You can have biosimilars, proteins which are present in humans, as well as they are also present in very similar looking proteins are present in bacteria. Essential enzymes like DNA, RNA synthesis enzymes has resulted in a lot of drug resistance coming. So you take the bacteria, you take different portions. For example, I was giving example of mycobacterium here. You can have cell wall, you can have the transcription machinery ones, you can have several other places, but you have so many options. So you can think of it that it's easier to design drugs for bacteria because your options are more, but it's easier to develop resistance because options are more. And with virals, the same problem happens. You have very few proteins that you have in your hand, and vaccines sometimes become very difficult to develop because the proteins mutate at a very high rate. So this brings us to this problem of antibiotic resistance, drug resistance. This is the silent killer which has been there. So we can all think of the pandemic which is going on, but then there are millions of people who are dying because they have become resistant to the first line and the second line and the ongoing lines of drugs. And every year we have been seeing more and more of this resistance problem, which is both prevalent in whether you go for virals or bacterial infections. So what my talk will focus today on trying to understand, suppose we are at a precipice and we want to look for new drug targets. What should we do? Should we look for new drug targets, or should we reverse resistance, or should we do both in parallel? So here I'm going to talk about a few of the directions from our lab where we have tried. I would not say we are at a basic stage. We have not reached a stage where everything is in the clinic. But how do I proceed with a new drug target? For example, you can discover new novel enzymes or pathways. I'll give you an example. You can also go and attack protein-protein interaction interfaces which are not conserved. For example, if you have a chemical reaction, it is more conserved. But if more than one protein comes together between species, just to make sure cross-species interaction doesn't happen, protein-protein interaction interfaces are not conserved. So you can kind of use them as drug targets. And in the last part of my talk, I would talk about origins of resistance, where in the previous slide, I did not mention many of these antibiotics are actually produced by streptomyces, and they not only produce the antibiotics, but they also have resistance mechanisms. So these resistant mechanisms are ancient. They're always there. It's a war between human and pathogen. How do they actually try to catch each other? So it's always going to be a war. We have to be one step ahead. So we are going to look at origins of antibiotic resistance with an example. So let me get to the first part of my talk, where I say, if I want to look for a new enzyme, so what will I do if I want to look for a new enzyme? I look for an essential enzyme. So it's very important to look for an essential enzyme, and it has to be different. It can be different function. Then it's sometimes difficult, because if it's completely different function, why is it not present in humans? It may not be an essential enzyme, but there can be cell wall synthesis and many other things. So we have chosen nucleobase deaminase as a target because there are many reasons. First of all, they're essential because they regulate this DNA and RNA pool, the bases, which are used for making these DNA and RNA, and they look different in both humans and in pathogens. So, for example, this is what I'm pointing out right now is a CDA fold, cytosine deaminase fold. It's called. It looks different. It's not present in the humans, and we have something called a triose isomerase fold, which this enzyme has, which is not present. So it's like having monkey and human, different looking looks, and from this looks, if we can come up with something, then we can use it. The reaction, let me tell you, it's the same, but the entry to the reaction or the way the enzymes look are different in these cases. So we chose this also because cytosine deaminase has already been used in the clinic. It's used as a prodrug where deamination reaction by cytosine deaminase happens, and this X is many times a fluorine molecule, and this is not toxic. The byproduct, which is the hydrolytic byproduct, is toxic, and you can target it with the various therapies, and you can use structure to make it more potent. And this has already been used for prostate cancer. So one family, a completely different application in cancers is already being used. So we thought, can we exploit this enzyme? So this is what I'm showing you, a sequence similarity network. And this is used many times to look for new enzymes. So we made a common ortholog group, COG, as it is called here, of this particular group. And what we did was we segregated the enzymes based on their sequence similarity. So any dots, like you can see these two green dots are two different proteins. Any of the dots, blue, pink, are different proteins. And how close they are given by these lines. So if they're very close, these lines are very short. If they're a different group, the way we have categorized them, they will be falling in different groups. Each group may have a particular function. For example, this group has adenosine deaminase activity, this has guanine deaminase. But what was striking is we were able to find a group of unknown function which was only present in Mycobacterium deaminases. So these enzymes are not present anywhere else. They're only present in Mycobacterium. So what are they doing there? They are going to be some kind of nucleobase deaminase. That's a given because the group has almost that function. But what kind of deaminase? Is it just an evolutionary different guanine or cytosine or cytidine or something else? So we were very, very perplexed by this, and we thought that maybe we can target them because they are in the essential group of enzymes. So first thing we did was we saw the crystal structure, the photograph of the enzyme. And this blue portion shows you what is common in all enzymes. It's a zinc-dependent enzyme. And the red portion shows you which is unique in this enzyme, and that is there on top of the active site. And so if you look here, this is our enzyme, MSD, and these are the other enzymes which are doing different functions, guanine deamination, deoxycytid delayed deaminase, and cytidine deaminase. So when you have a sugar group, there is some more embellishments added. So you can see this red portion is very different in all these CDA family members. The sad part was when we saw the structure was when we looked at MSD, this red portion did not look like any of the other red portions. So if you have a close-up of the enzyme active site, it did not look like any of these. So I can't just say that, okay, they were evolutionarily different, but this MSD was the cytosine deaminase, it was a guanine deaminase, I cannot tell. I can have a closer look at the catalytic site, and I can say it's like a hodgepodge between a guanine deaminase and a cytosine deaminase, but it looks like neither. So we were stuck. We had the photograph. We thought, photograph is a great thing to have, and we have the power, but that photograph did not tell us what the function of the enzyme was. We are able to tell most likely it's a deaminase, it's a zinc-dependent reaction. That's about it. So Vandana went about using, now how do you use the structure? We did a lot of docking of various nucleobases and to try to get candidate libraries. And we made a candidate library. And after a lot of these rounds of docking and adding more members to the library, we are finally able to tell that this enzyme does mutagenic bases like isoguanine is an example. It will a shuffled guanine position where NH2 and double bond have been shuffled is what it takes, and it's able to convert it to xanthine. So it will take this mutagenic base and then convert it into xanthine. Not only that, it would also take as a cytosine, which is right over here. So anything with 3 nitrogen atoms, like you can see in the ring, which is the more electron-rich system, it's an easier reaction than doing cytosine. So it would do that. It's and if you look at the active site, it is much more open. So it would do as a cytosine type of substrates and convert them, basically deaminate them. You get a double bond over here. So we solved the structure in this case to get better idea. And what we were able to tell is whenever you have the correct scaffold, only then it places itself in the correct orientation next to the zinc atom. And there is a water molecule which activates it, and this glutamic acid and this asparagine is very important for the reaction. And the reaction happens at the carbon adjacent to the NH2. There is a tetrahedral intermediate that is formed, and that's how this reaction happens. But the placement of the group is the most important. The minute the placement is not good, as you can see in this inhibitor, case of this inhibitor, you can immediately see that the placement of the group is no longer in the correct way. So just being able to look at the structure, we can tell what's a substrate, what is not. But a priori, we didn't have any knowledge. We were able to pick some of these, but still it was difficult. So yeah, we did a lot of mutagenic. What we did was we mutated all the bases, all the amino acids adjacent to the active site. And the question was to understand how important each and every amino acid is for the reaction. And what came out, whenever you lose activity, these bars, as you see, go down. So what came out is that if you look at the top pyrimidine-like purine-like scaffold, the activity goes down. The bars go down very fast. But in a pyrimidine-like scaffold, the bars are up for a long time. That means the pyrimidine scaffold is less sensitive than the purine scaffold. Maybe because the purine scaffold requires more shielding from the atmosphere, from the surroundings. And this reaction is shielded in a way that if this water molecule, for it to be activated by zinc, you should not have water all around. Most of the times, the active sites are very, very compact. So, so this is all very good. The structure was able to tell us a lot of things. We were able to tell, if you look here, what is a minimal scaffold. And this enzyme, what it does is it scans for certain mutagenic bases, and it does not deaminate any natural bases. So it's like this enzyme is like a scanning system. But we still didn't have a clue what it just did. And just before I end this portion, what I want to tell you is in Mycobacterium, this enzyme is like a gatekeeper. It looks like that. We need to do more studies for it, but it looks like it comes in a conserved gene pool where you can look at this gene area, and this is about export genes. A lot of export genes are present over here. So it's like, and Mycobacterium are resistant to triazine class of drugs. That means that if a triazine class of drugs comes into the Mycobacterium, it goes through the whole process of scanning it, and if turns out to be this type of scaffold, they're exported out of the cell. So Mycobacterium is innate immunity for these. Now, one thing, how can you use it as a drug target? you can maybe silence this enzyme. And we are trying to do it with the knockout studies now, and try to understand why, what happens. Do these compounds now become, suddenly mycobacterium becomes um, mm, uh, sensitive to triazine-based drugs, which it was known to not be sensitive. And that's what we are trying to do now, and see if we can exploit it to use it against organisms, and try to understand how uh, um, these kinds of protection mechanisms that are being harbored by these species. So the way forward would be to try to do more work in this direction. But I will leave you here thinking about this approach, where we can come up with a new enzyme out of a collection of enzymes, and show that this enzyme has potential as a drug target, and it will be specifically be used for only, for example, mycobacterium. And you know, in my first slide, first or 2 or 3 slides, I showed you how mycobacterium is a difficult target. And here you can have triazine-like drugs which are already there. Once you inactivate this enzyme, there's a possibility they can be used. So I will change gears now, and I will go to my next part of my talk. And here I will actually talk about the basics of this enzyme that I'm going to talk about, and later I'll show you how this may be exploited. But most of the talk will be about how protein-protein interaction surfaces or how ammonia channeling actually occurs, and can that be exploited as a potential arena where you can spend a lot of time developing drugs. And this work has been done mostly by Nandini, a graduate student in my lab, and this has recently appeared just on 3rd April. So I'll talk about what she has done and how this enzyme can then lead the way forward. Again, we have used an enzyme which is an essential pathway enzyme. That's very important whenever you're working with anything which is you want to use. You want to completely obliterate for example, pathogen in this case, not humans. So you would like to take an essential pathway. So here, this particular pathway is making of the purine bases, and if you can stop any of these enzymes, you are pretty much going to have no DNA and RNA being formed, and it's complete death. And these are essential pathways, so changing those pathways is also very difficult, because you don't want to affect the function. So that's another point. So, but very, very nicely, this pathway is different between humans and bacteria. For example, in bacteria, there are 3 different proteins which make up this pathway, and in human, it is 1 single polypeptide. It's in gram-negative bacteria actually, and archaea. So you can have 3 different proteins that are complex together to come together, and then you once they complex, only then you can actually get the activity of this particular enzyme. This enzyme is one of the steps, for example, in the purine biosynthetic pathway, and I'll show you which step it is in the next slide. But in humans, because this is a very important reaction, it is 1 single polypeptide. And however, the reaction this enzyme does is this particular reaction, which is it is trying to form this is 5-membered ring. So you have a purine base, so 5 and a 6-membered ring will be formed. So the 5-membered ring is being formed here, and glutamine gets converted to glutamate. Ammonia is channeled, and this ammonia that is channeled is then converted in an ATP-dependent fashion. C double bond O gets activated using ATP, and then you get a enamine. So this and then the 5-membered ring will be formed. So this looks seemingly a simple reaction, but it takes a very big enzyme to do this. And now I would like to stress that this was my PhD problem actually at Cornell, and my advisor said if you don't solve the structure of this enzyme, he will not give me a PhD. But he was joking, I hope. But I did solve the structure. This enzyme is very big. The green part is where the ammonia is produced. And this is a very, very big enzyme as compared to what it should be. Mostly bifunctional enzymes like this are about 50-55 kilodaltons. This is 140 kilodaltons. So this cyan and this blue color is a gene duplicated domain, and only one of them is active. This is the active site, and this is an ADP dependent site. This is all the work we have done in previous years. And this yellow domain is an adapted domain, and people don't know what it is doing. So even after, again, although I was trying to impress on the photography, only photography alone is not, it's going to tell you a lot, but it's not going to tell you how this enzyme functions. But the cool thing about this structure was that such a big structure, every part of this structure was actually ordered. The only disordered part, which I'll show you in the next slide, was this 22 amino acid. So if you imagine, if I don't know how many of you have done crystallography, and if it's protein crystallography, it's almost impossible to find a structure where the N and the C terminus is all ordered. And you can see these big loops over here are all ordered, and only one disordered part is there. And that's what Nandini encountered here. And this was right above the active site, this FGAR active site. So there are two active sites, like I mentioned. Ammonia will be produced here, and it will be somehow tunneled to this active site. And another thing to remember is, then this active site is completely dead in the absence of any substrate over here. So you will not get anything happening until unless you activate this site somehow. And this activation is happening when something is, the FGAR is entering here. So there is a communication 25 angstroms away that is existing. So how does this all happen is the question we said. Can then, once we understand this communication, this tunnel, we couldn't see any tunnel, we didn't know how it was happening. So we wanted to understand, can then we control the process to our favor? So we looked at this loop, and if you look in 500 large purelles, that is those which have only a single polypeptide, these regions are conserved. There's a lot of glycines you can see here. So this are the conserved regions. I've labeled them as a region which is close to the active site, which is where FGAR binding is happening. There's an interface. There is a region which may interact with the next domain, and then there is this extra region. So what Nandini did was she made many mutations, many cuts in pieces, and chopped this active site into pieces, and this loop into pieces, and made short version, long version, mutated versions. And what she saw was, if you may do anything in this blue region, it's just redundant. Nothing happens. You have ammonia production. Nothing happens to the enzyme. But if you take this yellow, this golden, and this red portion, you can see here, as you make the mutation, you do have ammonia production happening. That means communication between active site is happening, but no FGAR is being produced. That means something has happened to this site. This site is still functional. Okay, but you look at these black ones. As you make the mutations in the black ones here, you see that slowly ammonia production is lost, and because ammonia production is lost, you see almost loss of activity. Now this black has nothing to do with binding with the substrate. It is only supposed to contact the next domain. Okay, so so that was pretty perplexing, because 30 angstrom away you are trying to make mutations here, and here the activity is gone. It cannot process ammonia anymore. So that was a little bit surprising to us. So what we did was, at this point, at this juncture, is that we made slow mutants, and I'll race through this so that I can go to the last part of my talk. But we raced through, we made slow mutations, and immediately after we made the slow mutations, we were able to trap the enzyme for the first time. It was very difficult to get the loop in the proper orientation where you have the FGAR binding happen, and there is glutamine in this active site. And using this orientation, we did contact our Jagannath Mandal, who is our collaborator for simulations. And he did a 1 microsecond simulation, and we were able to show very efficiently that this particular protein has a lot of interaction. So to make a long story short, you can see these portions which are this N terminal, this domain, which is the yellow domain, suddenly shows a lot of motion, and this loop, which is in red, is showing a lot of motion. And if I show you this morph movie, as the loop opens, it hits this domain, and then this pushes this domain. Okay, and this is how the activity most likely happens. To prove this, whether this is really happening or not, and it's just a fiction of a simulation, what we did was we took this this loop interactions and studied them. So you can see this is before the loop was ordered, and this is when the loop was contacting the half open form of the loop. And what we did was, we had already made mutations in the loop part. Now we did not make any mutations in the loop part. We made all the mutations in the yellow part. So you can see, when you make the mutations in the loop part or the yellow part, completely glutamine binding is gone, and the same result happens. So whether you make mutations in the black part or the red part, which is the contracting domain, you see exactly the same effect. So this tells you that really the signal is being passed from here to here, and this is an allosteric switch. And once the signal is passed to this site will become active for accepting glutamine. Otherwise, it cannot even bind glutamine. So this was a good discovery for us, because now we knew the origin of the allosteric switch. We went and also looked at the changes over at this site, how this activation happened. And it turned out that it looks like that this activation happens because lots of interactions, this yellow and this green domain and the FR domain breaks, and new interaction, it kind of frees something called an oxygen ion loop and makes this this particular region come to its active form. Not only that, it opens up a passage through which ammonia can now, glutamine can now enter. So all these motions we were able to see, and even the icing to the cake was that we were able to see the whole channel. So nobody knew how the channel was existing so far. We were able to show how the channel was being formed. We were able to show that there were two gates and how the gates open, like histidine, and we can see here also another arginine, asparagine, glutamate gate over here. So two gates were there where ammonia was coming out, and I'm not going to tell you details here, but basically I can share with you if you want, but in summary, we were able to show the functioning. But then what is, we have learned about the allosteric switch, we have been able to find the channel, we have been able to tell how this is happening, but how does it help in drug development? So this protein-protein interaction surface, I said, remember 3 proteins came together and not single protein? This protein-protein interaction surface is completely different in bacteria and archaea. And this is then in humans. So you can kind of avoid this interface from being formed actually, and not disrupt the human interface, because they are completely different. They are species specific. So you can see that in large purelles, they're conserved in one way, and in the small purelles, as we call it, the 3 different protein complexes, they are different. So this is where you would target therapy, because you can make protein, you can prevent these proteins from interacting, because the amino acids are completely different in both the sites. With this, I will leave you, and I will come to the last part of my talk. Sorry for burdening you with this part, but I really do want to talk about this part, because this is telling you that resistance is prevalent everywhere, and it's present. And can we combat this resistance? And we don't need to develop any new drugs. If we can just stop this resistance from happening, then we are kind of safe. So this work was done by Ruchika in my lab, and a part of this work has been published, and I'm going to talk about this work. And the problem that we are addressing here is, like I said, origins of antibiotic resistance. So many times when bacteria want to attack, what they do is they attack by stopping protein formation. So that is being done by this giant molecule called the ribosome. And here in the ribosome, each and every amino acid comes in a controlled fashion, and you get via tunnel, things come out. So what bacteria do is they attack many places, but one of the places they attack is the tunnel itself. So in this tunnel, what happens is this macrolide goes and binds, and when this goes and binds, no longer bacteria can actually form their proteins. We don't, the way the bacteria get resistance is they do not like this macrolide to bind. How they do it? They actually use a bunch of enzymes called methyltransferase. Very, very elegant reaction. All these drugs stop working. So if we can stop this methyltransferase reaction from happening, then we can actually develop drugs. And again, these macrolides can be used. This is a very common way. Many of the common erythromycin, streptomycin, and the drugs that you use daily are becoming extinct because of these modifications. And there are many such modifications which happen on the bacteria. So if we understand the origins of these modifications and how we can stop them, we can know a lot about trying to design combination therapies where we can just stop this enzyme. So what is the modification? On the adenine, which is present in the bacterial ribosome, we don't have this. We have a guanine, and that's why we are safe from these macrolides. A methylation, mono or dimethylation happens. So once this methylation happens, no longer the drug can bind, there is a steric clash. So because there is a steric clash, now the macrolide cannot bind and bacteria can happily do their synthesis, okay, of their proteins. So we want to know, can we stop, and how do the bacteria methylate, first of all? How do they know they have to go to this A2058 position and nowhere else? Because they just don't randomly methylate anything else. They only methylate the correct position. So how do they selectively methylate? How do they, can we stop this process if possible? And if methylation doesn't happen, our drugs will work. So we don't have to come up with new drugs at least. So what Ruchika did was, we said, okay, let me try to figure out how this particular enzyme knows it has to go to this site and not here. So she took an enzyme which went to this site and not this site and said, can I make this particular methyl transferase, which is a surface methyl transferase, become like a pathogenic methyl transferase? So what she did was, the question is, how does bacteria know where to go? And actually this protein has this embedded information. Again, the question is clear, but if you look at both the enzymes, they look very similar. So you must be thinking, this is called the Rossmann fold, this central portion, and this is called the head domain, which is slightly different between the two, but the basic catalytic site, which is F-adenosine methionine based, is absolutely same in both, and it's conserved. So you ought to think that how does this enzyme not mess up, and it will only take surface methyl transferase substrates, and this will be a pathogenic enzyme. So Ruchika wanted to convert this enzyme into this enzyme, because what it looks like is, it is a recognition of the RNA, the ribosomal RNA, which is different between them. If in this small protein, this information is embedded and we can take it out of it, we can get a lot of things about this enzyme. So Ruchika did that. Ruchika went ahead and solved the structure of this methyl transferase with and without F-adenosine methionine. And she looked at regions in the enzyme which are moving a lot when this F-adenosine methionine enters. Why we are doing that? Because we want to look at places like this lysines, K24, K23, which are positively charged because they're inviting the RNA. So if we see activity changes or RNA binding differences, then we can kind of know if this is the region where RNA is trying to come in. So, so this loop looked like one of the regions where RNA may be interacting with. We looked at all the other structure, and what Ruchika did was she made a phylogenetic analysis. This is the red portion is the urn, the pathogenic, blue is the starting enzyme. We want the starting enzyme to become like this. And if we make various cuts and pastes, like, differences in structure, differences in their where the sequences don't align that well, and if we can make many such chimeras and actually make theoretically and see what makes the ancestry change, what makes them go and to become ERM, then it will be good. Not only that, this is in silico. What Ruchika did was actually made them. She made all these. The aim was this pathogenic template RNA, which is present in, and this site is there, which is responsible for methylation and then resistance. This particular enzyme starts working on it. Because then we will know what are the RNA, what are the targeting determinants, and what are the parts of the protein which are recognizing the right RNA. So she made many chimeras, and to make a long story short, she was able to show that if you just swap two loops, then it becomes a pathogenic enzyme. Not only that, we compared its activity with other pathogenic ERMs, and you can see that it's 50% as active. We have not got 100%, but it is acting like it under these circumstances in the test tube. So you are at the test tube level right now. So we decided to go and put this particular enzyme as a plasmid and go into the organism itself, which was an E. coli cell, and it was sensitive to erythromycin. So when you add the original one, it will suddenly become resistant. And if you add the chimera, it turned out this chimera would work not so well. But if you also change the head, it worked really well. So what I'm trying to tell you here is you have a combination of changes and it becomes a pathogenic enzyme. In the test tube, these two loops are enough, but in the cell where it has to find the entire ribosome, the head is also important. So this head seems to be playing a very important role in targeting. And we try to understand its function a little bit more, that how you proceed from the cell to the template to the cell stage and what has become important. And we solve the structure of the 30th ribosome of the surface one, KSGA, and try to understand how the various portions, how is it trying to find its position in the ribosomal context. And this we have done in collaboration with Dr. Vinod Kumar at NCBS. And what we find out here is that many portions of the enzyme, the head, for example, and the loops that were important are interacting with peripheral regions, not the actual active site. And they are presenting the actual active site. So the interactions are happening as a combination of the head in the loop with the various parts of the ribosome. So it's like a scanning algorithm, which is there inside this protein, and it is trying to match it so that it is going to its correct active site. So if we change these things, the algorithm has shifted and it has become a pathogen one. So you don't have to change the reaction in any way. But then that's very good. We have been able to do a lot of these things, but how do you now design a drug with it? You can still look at the RNA sequence very carefully and see how is it actually interacting. And it is a 31 angstrom interface between the protein and the RNA. And within this 31 angstrom, there is this region, which is the very, very conserved region of the RNA, which is interacting with the enzyme. And this is a ribosomal RNA. And you can see that we had done a bunch of experiments, and we found out that these bulge regions over here, you can see ACG, this bulge, are very important. So here is the active site, and the bulges, 2058 is here, and these bulges are over here. And these bulges seem to play a role in trying to recognize the RNA. And each bulge and protein interface is unique. For example, ERMS has one unique combination, and the KSG has its own unique combination. These bulges will be in a different place in ERM and a different place in KSG. And by changing the loops, we actually enable them to recognize these bulges. So when we were making these chimeras, we were actually changing the loops and the regions. And so the RNA and the bulges it was recognizing became different in both cases. And what that tells, to study that even further, we had decorated the RNA site specifically with aminopurine, with thioguanosine, and tried to see the conformational changes. For example, this study at the level of just the active site has been done in DNA methyltransferases, where every time they are kind of stacked, and an unstacking event means methylation. Unstacking event means increase in fluorescence. So a lot of change in conformation also means increase in fluorescence. So we took this template, we wanted to do in RNA methyltransfer, what are the conformational changes that are happening? And what we see is, yes, A205, it is kind of flipping or changing conformation because there is an increase in fluorescence. Not only that, these bulge sites, let me show, I'll skip it in the interest of time, but these bulge sites are also showing huge changes in fluorescence when the protein is coming near it. So what is happening? So in summary, what we think is happening is a simulation experiment we did in collaboration with Dr. Naresh Patwari's lab. And what we here found out was that the whole RNA, it's a protein RNA handshake that happens, and lots of motion happens. And this is my last slide. It basically tells that the RNA actually goes through a dramatic motion. So, and these bulges that I was talking about and these loops, they interact with one another. They form unique interfaces. These unique interfaces are very important because this unique interface is 10 angstroms away from the site. So this is where the reaction happens. This is where the bulges are. So let me show you. These bulges interact with these this region, this loop, which is the loop I told you, the chimerized loop, and the other chimerized loop is here. They are interacting here also, and these changes percolate down the length of the of this whole thing, whole interface, and you ultimately get this is the active site of flip happens. So what I'm trying to say here in short is you have 25 angstroms away or more changes happening over here, and you will see the reaction happen on a reaction is facilitated at this end. You remove this bulge, no reaction. So if I want to design drugs, this is the site I would choose, because this site, all S-adenosine methionine methyl transferases are going to choose this, and you will have side reactivities. But this site is unique for your pathogenic enzyme, unique combination, and that's what we are doing now. We are trying to develop various kinds of strategies. We're trying to do small molecule aptamer based, because very positively charged region, to be able to have a unique, against this unique cavity, develop molecules which will be allosteric inhibitors, and they will not attack the actual active site. As you can see, this particular lock and key of the RNA and the protein is only present in this particular pathogen. So it's like a unique pathogen RNA system, and that's how the recognition happens. So you can design this without any side reactivities, and that's what we are trying to pursue right now. And so in short, what I tried to tell you today through my presentation was various strategies. I'll just recap. In the first one, we discovered a new enzyme that was never known to exist in Mycobacterium tuberculosis, which was a scanning enzyme for pathogenic bases. It would only deaminate not pathogenic bases, mutagenic bases. So it was preventing any mutagenic bases to reach the DNA and RNA. And if so Mycobacterium will be more resistant to any mutagenic bases. Why? Because they're never able to reach them. That's why. It's only exporting them out of the cell. The second thing I talked about is a whole mechanism which is in a purine salvage pathway, that is forming purine bases, essential for DNA and RNA synthesis. Here also, instead of attacking the active sites itself, we were looking at protein-protein interaction interfaces, which are different between humans and gram-negative bacteria. So you can use that as a leverage against you. To do that, however, we had to first understand the entire system, figure out where the allosteric switch is, what kind of place we should then concentrate on. And the last part, I talked about reversing resistance completely. And here I talked about this small methylation events that happen on the ribosome. And these methylation events happen on the DNA also. They silence the DNA, RNA splicing. This methylation, methyl transferase is like a ubiquitous enzyme which is specific yet prevalent. And in this case, this methyl transferase, which is an epigenetic modification, is prevalent everywhere in nature. And it has shown here that you can actually use this methyl transferase. You can block this methylation. You can then come up with inhibitors. With that, I'm going to end my talk. I've thanked the people. I thanked them along the way. We had different collaborators for different parts of the talk, and my collaborators for the last part, Eve Smelly helped us with the stock flow. Professor Naresh Patwari helped us with the last simulations, but for the ammonia channeling, it was Jagannath Mandal's group in TIFR which helped us, and Vinod is still helping. We are doing a lot of work with actually dealing with the cryo-EM structures now, and trying to understand in a ribosomal context. We are mutating the ribosomes and then doing the same study. That's a large activity which is being produced, which is a future activity being pursued in our lab. I would also like to thank all the agencies, and I would like to thank again ACS for giving me this opportunity to present my work, and thank you all for listening at my high speed barrage of discoveries. I may have gotten a little bit more, involved in it and very fast at some point. I have that problem. So I would be happy to answer any questions, and stay safe and healthy. Thank you.